Good afternoon, everyone. I hope you all had a great lunch. I'm sorry we weren't able to buy it for you, but we will next time when hopefully we meet in person. I hope you've had a great morning. I think the range of talks was absolutely fantastic. From Eve to React to Rwanda to recovery to open safely to care homes to cancer. I should say that all content will be made available freely from ourselves and our partners. So if you've missed any sessions, you can catch up. Also on questions, I think it was Phil Quinlan flagged that it'd be really good to see the questions. And I understand now you'll be able to see the questions from all the sessions on the message wall. Anyway, let's get on with the plenary. A huge pleasure to welcome Adam Butterworth, who's a stalwart of Health Data Research UK in Cambridge. It's precisely 6,646 days since the first human genome was sequenced on April the 14th, 2003. And now we live in a world of omics, whether it's genomics, proteomics, lipidomics, metabolomics, or the most important one, economics. So it's very important that we think, how will the omic revolution interface with the phenotype as derived from electronic health records? So Adam, you're going to tell us about the future. A huge welcome. Please put your questions in the chat. We're going to try and get some questions at the end. But Adam, take it away.
Thanks, Andrew. It's a privilege to speak at such a fantastic event and a challenge to follow such incredible lightning talks before lunch. So I'm glad no one's voting on my talk. As you said, I'm going to tell you about the opportunities to capitalize on the UK's excellence in several areas of population science, the genomics that you've mentioned and the incredible revolution we've seen there over the past 20 years. The amazing health data that we have that enables us to look at all kinds of diseases and health outcomes, and specifically about the molecular factors in between. And you've mentioned some of the many omics that have been measured in population studies in the UK and beyond that we're trying to use to enhance our understanding of the causes of disease. And of course, that's one of the central themes from HDR UK. So I'm going to give you a flavor of the kind of research that's going on under our From Molecules to Electronic Health Records badge. And there are three main strands of the work in that area, which I'll take you through and give you some examples, including some related to work that's gone on in the last year relating to the COVID pandemic. So first, multiomics cohorts. I would like to use a very recent example to illustrate the paradigm here. And this is work that's been jointly between the University of Leicester and the University of Cambridge using the UK Biobank, the national flagship population cohort. Our colleagues at the University of Leicester spent several years measuring telomere length in all 500,000 people. And for those who don't know what telomeres are, they're the protective ends of the chromosomes of DNA. As we age and our cells reproduce, each time we lose a little bit of DNA. So we have these repeat sequences of kind of nonsense DNA at the end of our chromosomes that don't have any genes in so that we're protected. And there's been therefore great interest in trying to measure telomere length to see, is it a marker of how well or badly we're aging, and what's its relevance to disease? So we were able to link this telomere length measurement with the health record data in UK Biobank and look at the observational associations that you see here on the left between different telomere length and different diseases. And you see, for example, there are positive associations with several types of cancers. There are non-linear relationships with some diseases like leukemia, and even inverse relationships as seen there with liver cirrhosis. But of course, these observational associations may well be confounded. They may well be consequences of disease processes rather than causes. So what we really want to get to in the understanding causes of disease theme is some element of causality. And of course, the way we try to do this is by linking in genetic data. So because we have genetic data in UK Biobank across the whole genome, you can see here we can then interrogate that with a particular risk factor like telomere length, and in this case found nearly 200 different regions of the genome that seem to influence an individual's telomere length. And then, of course, we can try and combine this all together. We can bring together the genetic data, the telomere length data, and the disease outcome data to try and understand which of these observational associations might be causal. And again, here's a snapshot of some of the ones that looked interesting. For example, you can see here that some of those cancers I showed you on the left-hand side of the screen, those observational relationships do appear to be causal. But there are associations also in the opposite direction with things like coronary disease and the liver cirrhosis that we saw. So we can take one risk factor and look at its causal influence on a wide range of different diseases. And in fact, just today, we've had the good news that another paper in this regard has been accepted in eBiomedicine, looking at the relationship of telomere length in UK Biobank with COVID-19, suggesting that there does seem to be a relationship, even after we account for chronological age and other potential confounders. So some further work there to be done to try and understand potential mechanism of association. Now, of course, as Andrew mentioned, we're no longer thinking about single risk factors, but there's been a revolution in omics that allow us to measure many thousands of risk factors. And so rather than just looking at the genetic determinants and the causal consequences of one factor like telomere length, we've been starting to try and do this with many thousands of proteins, as exemplified here. This was data looking at about 3,500 proteins and finding nearly 2,000 genotype to protein associations, giving us this incredible catalogue with which to interrogate causal pathways. Now, those were both results from individual cohorts, and as I've mentioned, one of the things we wanted to do was to try and capitalize on the UK's great strength in this area. And so I was lucky to be funded by HDR UK to lead a program that we're calling the HDR UK Multiomics Cohorts Consortium that brings together different cohorts from across the UK that you can see on the map on the left, from the Scottish Isles up in the north, down to Avon and Bristol down in the southwest, over to Norfolk in the east. Currently around a dozen cohorts with about 800,000 participants who've all given their consent for blood samples to allow measurement of DNA and these other omics molecular layers, as well as for linkage to health records that allow us then to look at the disease outcomes. And of course, the aims of this work are really to integrate those layers of data, as I showed you for the telomere length, to try and answer several different questions. So to find causes of multiple different diseases, to try and prioritize different targets for therapies and inform drug development, and to also think about things like predicting risk of future disease as well. So one can then think about how we can do this when we combine data from several studies. This is an exemplar led by Chris Yang in Bristol in an academic industry collaboration that was with heavy involvement from GlaxoSmithKline. And we can start to say, well, rather than looking at one risk factor or one disease, what happens when we look, for example, here at nearly 1,000 proteins and their potential causal effects on more than 200 different diseases and traits. And we can start to build this atlas of different causal associations across a wide range of different diseases that you see here in the different colored dots and a wide range of different proteins and pathways. Now, of course, a year or so ago when the COVID pandemic hit, we wondered whether or not these methods might be suitable for a new infectious disease like COVID-19. And several different cohorts within the consortium tried to enhance our understanding in this area. This is an example of work led by Mike Pietzner and Claudia Langenberg at the MRC Epidemiology Unit, who identified a subset of proteins that were thought at that point to be relevant to either interactions with the SARS-CoV-2 virus or response to COVID-19 on the basis of biological pathways like coagulation or cytokine storms that we knew at the time. And they were able to find genetic signals for about 100 of these proteins and to make them immediately and publicly available so that the world could start using this genetic information, the genetic links between these variants and proteins to try and interrogate COVID-19 further. One way in which we did that was led by Liam Gaziano. You see up here on the right, who's a joint international PhD student between my lab in Cambridge, UK, and Juan Pablo Casas' lab in Boston in the US. He was able to take these genotype to protein findings as well as genotype to gene expression findings that have been published previously. And he was able to then try and integrate that with emerging data from around the world, linking genetics to COVID-19 outcomes. And what you see on this slide is then an illustration across the whole genome of the different areas that lit up for us, the protein pathways that we thought might be relevant to COVID-19. We were heartened to find that some of the results made a lot of biological sense. So, for example, one of the findings was here on the X chromosome at the right-hand end, the ACE2 gene, which is known to be the receptor for the SARS-CoV-2 protein, virus rather. The second example that was we focused on in this paper was an interferon receptor, and again, which had high biological plausibility and points towards a certain class of therapeutics which may be useful in early COVID-19. So we were able to take the same methods that we'd been working on across a range of different diseases and start trying to apply those to COVID-19 in a rapid time frame. So that's a whistle-stop tour of the multiomics cohorts. I'd like to tell you a bit now about another main strand of the work, which is about building tools and resources for the community, which is clearly a core focus of HDR UK's work. We're building here on examples like our Phenoscanner database. This was the idea that we were, as you saw there, we were creating so many hundreds or thousands of genotype to phenotype associations. We needed central repositories and places we could interrogate them all. And so we built this database to do that. It now has about 65 billion of these associations in it, and has been widely used, as you see there on the right, in many papers by many investigators from around the world to try and understand or to try and synthesize what we know about the relationship between the human genome and these molecular factors, and to use it to try and infer causal relationships. More recently, we've then been trying to do this in perhaps the next frontier in molecular science, which is an area that you may have heard about called polygenic risk scores. For those who aren't familiar with this, the idea is that we can move beyond what you see on the left here, which is individual genetic variants that seem to have some kind of association with a phenotype or a disease risk, and start to aggregate variants together in the hundreds, thousands, or even millions. And the idea here is that although individual variants may often confer relatively small changes in disease risk, once we aggregate them together and understand the bell curves, if you like, that exist in the population, we can often then see a difference between people who have a disease and people who don't, be that coronary heart disease or lung cancer or even COVID-19. And what we can start to then do is look at each individual and say, were you at the left-hand end of this curve? Did you get a lucky genetic hand and have a low risk of disease? Or are you at the high end of this and you got the unlucky genetic risk? And of course, we can see if that's translatable into anything that might be clinically meaningful. This is an example of work led by Luan Sun and Emanuele D'Angelantonio here in Cambridge, looking at coronary heart disease, where we have screening programs using existing risk scores. And you see from the data in the middle that if we were trying to predict coronary heart disease, we can get good prediction using a traditional risk score. This is in UK Biobank, so it's integrating information on traditional factors like smoking and cholesterol, etc. But if you add a polygenic risk score component to that, we can get quite a substantial improvement in the prediction, and even more so than other markers such as C-reactive protein that have been proposed to improve these scores. And as you see from the quote from the paper there on the left, that actually once we use GP data from the GPRD and try and understand what the benefit could be at a population level, we estimated that this could prevent around 7% more cardiovascular events than we do at the moment if we could somehow find a way to translate these polygenic risk scores into clinical practice. Now, most of the research on polygenic scores has focused on predicting disease, but actually we've started to try and take, again, a look at these multi-omic analyses, these molecular risk factors, and say, could we use polygenic scores to try and understand causes of disease? And this is an example here from Scott Ritchie and Mike Inoue, who've been looking at combining proteomics. What you see here in the right panel is a polygenic score for type 2 diabetes, and then protein associations with that polygenic score. So can we distill down from the thousands or millions of variants that are going into that polygenic score some novel pathways that are causing the diseases you see here that could then potentially be intervened upon? So initial insights that actually polygenic scores may not just be good for predicting disease, but may also give us etiological insights that we weren't getting from the individual variants. Now, of course, when you have a new and emerging field like polygenic scores, then there are often gaps in the evidence and gaps in the tools and resources. And so a recent initiative sponsored by HDR UK and done by the same team at the University of Cambridge, including Sam Lambert, as well as Jackie MacArthur and others at the EMBL EBI, an American genomics institute, that have built a tool called the Polygenic Score Catalogue. And you see here, as of yesterday, there are now 800 polygenic scores that are included in this. And the idea here is that it will firstly capture all the published polygenic scores in one place, so making them accessible and findable. But it will also include the relevant data and software to allow benchmarking of these different tools. How do we compare them? Multiple people are deriving their own different polygenic scores on different data sets. How can we work out which ones are better, which ones are most suitable in certain situations? And of course, importantly, to do that, what we need is the metadata. And we've heard already in several of the sessions this morning about the importance of collecting metadata and the importance of reproducibility. So each of these scores has information. You may just be able to see, for example, on the bottom left here, it tells you that in this score there are about 1.7 million genetic variants. And perhaps more importantly, on the right, it tells you about the ancestry of the participants who are included at each of the different stages of deriving this score, so that we can start to address one of the key challenges of polygenic scores and introducing them into clinical practice, which is the enormous inequity in the performance of these scores across different backgrounds, different ethnicities, because of the wealth of genomic data that's emerged in European ancestry participants. And so how can we try and improve on that in going forwards in non-European ancestries? Importantly, as well, of course, for a new field, we need reporting standards. At the moment, it wasn't clear exactly what papers that look at these polygenic scores should be presenting. How should we be rigorously comparing these? And again, the same team who brought you the PGS catalogue have been involved in an international effort driving standards and publishing this paper in Nature earlier this year to kind of set the stage, to set the field to a level where we can now start to try and ensure that methods are reproducible and well reported. So finally, in the couple of minutes I've got left, I'd like to also touch on the third strand, which is about empowering genomic medicine. And we heard from Sharon Peacock and others this morning about how the genomic genie is out of the bottle. We are using genomics in some aspects already in the clinic, and this is going to be an exciting area, I think, of the understanding causes of disease theme going forward, particularly as we think about how to link genomic variation, multi-omic molecular data, and electronic health records in patients and patient cohorts, as well as the population cohorts I showed you earlier. This is just one really nice example that's recently emerged from Joanna Kaplanis and Matt Hurls at the Sanger Institute. This is based on data from the 100,000 Genomes project from Genomics England. And what we're looking at here is what we call mutation rate. So we know that having a high number of mutations in your genome that have arisen spontaneously is associated with higher risk of diseases, and they were particularly interested in here in developmental disorders. And we also know already that the number of mutations that you have in your genome is correlated with the age of your parents, and you see that in the kind of grey clouds on the left for paternal age at the top, but also to some extent maternal age at the bottom. Now, in population science, we often tend to look at populations at averages, and we often tend to exclude outliers because they can divert our models, or they can we throw them away because they're not where the bulk of the data are. Of course, in clinical medicine and genomic medicine, we are interested in those outliers. And so what Joanna and Matt and the team at the Sanger nicely did is to identify a set of around a dozen individuals who had an extremely high rate of mutation in their genome. And rather than just asking how high the mutation rate is and what the consequences of that might be, they were interested in understanding the causes of that. What can we learn about why this happens? Well, some of this we can just get from the genomic sequence data by having the genomes of the offspring as well as the two parents. They were able to identify that 4 of these were probably due to genetic reasons. However, by linking with the health record data and exploring in detail, they were able to identify that 5 of these individuals probably had extremely high mutation rates because their fathers had had cancer and then chemotherapy prior to conception of the child. And so particularly two classes of chemotherapy that seem to give rise to distinct mutation signatures and these high rates of mutations, with potential consequences, of course, for reproductive choices, particularly for men who are unlucky enough to have cancer at an early age prior to having children. And of course, many more examples of those that could hopefully emerge in the coming years with the proliferation of linkage of individuals with genomic data, multiomic data, and electronic health records. So I hope I've convinced you that linking together genomic, multiomic data, and health record data is enormously powerful in a range of different ways. Thinking a bit more about the future, and I've alluded to some of the items that I think we might want to consider in future, but three points that we will certainly be considering. Firstly, enhanced diversity. As I alluded to, much of this has been done in if you like, white European ancestry participants. So capturing more of the breadth of the participants across the UK will be important, as well as partnering with others around the world, such as in Bangladesh, China, Mexico, and the US, as you see here, to try and understand how generalizable our results are, but also to make sure that when we do things like develop tools like polygenic risk scores, that we make sure that they're sufficiently powered and usable in all different walks of life. Secondly, deeper data linkages. From the initial data linkages we have, we've obviously been able to mine an important amount, but working with initiatives like the British Heart Foundation Data Science Centre, led by Kathy Sudlow, and NHS Digital, we want to extend those linkages to delve deeper into diseases and broaden the range of diseases we can look at. And finally, of course, multimorbidity. We've heard this morning already about how COVID-19 hit worst among people who already had multiple different common diseases. How can we use our tools to go beyond one disease at a time approaches to look at constellation of diseases? So, of course, we must thank the enormous amount of funders, not only HDR UK that's made this all happen, but the academic and charity funders on the left, the huge number of industry and funders and assay providers on the right and at the bottom who've paid for a lot of these hugely expensive multiomic measurements in these population cohorts. And of course, I don't have time or space to list everybody on the slide who's contributed to the understanding causes of disease theme, but particular thanks to the Multiomics Consortium and the Polygenic Score Catalogue team, as well as, of course, to all the patient and public panels that we have and our representatives on the steering committees who are helping us co-produce the research. And of course, the study participants there, many hundreds of thousands of them who've all given their informed consent to allow us to take the DNA measurements, the blood measurements, and link to their health records to make this work happen. So thank you very much, and happy to take questions if there's any time.
Adam, we've probably got time for two very quick questions. So fantastic. So first of all, a question from Kit Byatt. So how much do epigenetics affect the accuracy of predictive models and propensities for disease derived from their genomes?
Fantastic question, and that's one of the ones that we really need to explore further. We've had a focus initially on proteins and metabolites, which are perhaps most commonly measured across the 13 population cohorts I showed you, but actually extending to other domains of multiomics is hugely important. We know that some of these cohorts do have epigenomics, particularly methylation, and so looking at that would definitely be a key thing to answer in the next few years. Yeah.
Okay, and very quickly from Alex Handy, and I'll paraphrase this. So if I was in my diabetes clinic in 10 years' time, Sharon today said that viral genomic sequencing had crossed the Rubicon. What tools do you think I will be using to influence decision making?
That's an excellent question, and I was at an American meeting last week that was trying to answer exactly that, and somebody said, why have we had these multiomics tools now or platforms for a few years, but they haven't made it through to the clinic? What do we need to do to follow the genomics revolution to turn some of these other omics into clinically useful? I think some of that is in improvements in the way that we measure these. I think a lot of that is in extensions in the way that we study these. So I've shown you a lot of information about associations and potentially causal associations, but I think taking that to the next step in terms of trying to show the clinical utility, as I mentioned, for the polygenic risk scores, what's the risk benefit, what's the cost effectiveness, I think those are some of the areas we need to move into more to the implementation science if we're ever going to realize some of these high dimensional omics into the clinic.
So it comes back to economics. Okay, we'll have to leave it there. So Adam, on behalf of the team, can I thank you and your team for outstanding work and a great plenary session. So the customary round of applause. So thank you.
Thanks very much.
And now, folks, I'd encourage you to log on to your favored parallel session because there's some great stuff happening right now. So thank you very much.